Hello everyone. Myself Mr. Samir Pawar, working as a assistant professor from Ganba Sopanrao Mose College of Pharmacy, Wagholi, Pune, and I am here going to deliver a short seminar on topic antibiotics. This topic is first chapter of syllabus of final year B Pharmacy as per Savitribai Phule Pune University. So let's begin with the chapter antibiotics. Here we are going to discuss the definition, characteristics of antibiotics and classification depending upon the different ways or basis of antibiotics. Let's see one by one. The first one is the definition of antibiotics. Antibiotics are defined as chemical substances or compounds produced by various species of microorganisms such as bacteria and fungi, which in low concentrations destroy, kill, or inhibit the growth of other species of microorganisms. It is actually Greek words which split it into two words, that is anti means against and bios means life, that is antibiotics. So these are the chemical compounds which are produced from microorganisms, and that metabolic products are utilized to kill, destroy, or inhibit the growth of other species of microorganisms. The next is the characteristics of antibiotics. These are the properties of antibiotics or the ideal properties of antibiotics. Let's see it one by one. So first one is it should have a wide spectrum of activity with the ability to destroy or inhibit many different species of pathogenic organisms. As we know that antibiotics are the metabolic products which are produced from microorganisms, and that are utilized to kill or inhibit the growth of number of species of microorganisms. So antibiotic should have the maximum capacity or wide spectrum to kill or inhibit the growth of other species of microorganisms. Next, it should be eliminated completely from the body. After showing its action, it should not remain into the body, as it will produces the other kind of side effects or adverse effects towards the body. So it should get eliminated completely from the body. It should not produce adverse and side effects, as no any drug will shows these ideal properties. So we can say that antibiotics should have or should show some little or minimum adverse or side effects. Next, it should antagonize the growth or survival of one or more species of microorganisms. Next, it should be highly effective in low concentrations. Means we can say that the antibiotic should be potent one. Next property is it should be non-allergenic to the host. Means the antibiotics where we are utilizing into the body should not produce any allergic reactions such as rashes, fever, inflammation, or any other severe cases. It should not eliminate the normal flora of the host, as we know that in our body some kind of the natural flora of microorganisms are present. If we take the example of gastrointestinal tract, there are some bacteria which releases enzymes and help in digestion process, and this shows its their synergistic growth also. So antibiotics, as we know the definition, they kill or inhibit the growth of that microorganisms. So antibiotics should not eliminate that natural flora which will affect the physiological processes into the body. It should be able to reach the part of the human body where the infection is occurring. Means the drug should reach, that is absorbed and get distributed properly where its site of action. It should be inexpensive and easy to produce. As antibiotics, we can say that one of the fermentation process by which that antibiotics are produced from microorganisms, so that process should be inexpensive and the antibiotics should get easily obtained from that microorganism or from that procedure. It should be chemically stable, that means it have a long shelf life. It should not react with the excipients or any other material to which it will get in contact. It is a product of metabolism, means it is get obtained from the microorganism. So these are some characteristics of antibiotics. Let's see the classification of antibiotics. So there are number of ways the antibiotics are going to be classified depending upon the different basis. So let's see one by one the different classifications of antibiotics. So first, antibiotics are classified depending or based on the source from which the antibiotics are going to be obtained. So depending upon the source, the antibiotics are classified as natural antibiotics and semi-synthetic antibiotics. So natural antibiotics, here are some examples such as penicillin, which is obtained from microorganism, the species of bacteria Penicillium chrysogenum, bacitracin from Bacillus subtilis, polymyxin from Bacillus polymaxa, streptomycin from Streptomyces griseus, chlorotetracyclines from Streptomyces aureofaciens, erythromycin from Streptomyces erythrus, and chloramphenicol from Streptomyces venezuela. Where semi-synthetic antibiotics are commercially synthesized by adding some chemical compounds or amides etc. to the natural antibiotics. Means here with that adding compounds get as a side chain into the natural antibiotics product. In these, the main part of the chemical structure is obtained from the microorganisms, which are then modified by adding various chemical moieties as the side chain. So examples of semi-synthetic antibiotics are ampicillin, amoxicillin, oxacillin, cloxacillin, dicloxacillin, carbanicillin and methicillin, etc. The next class where the antibiotics are classified, it is based on the antimicrobial activity. So depending upon to which microorganisms the antibiotics kill or inhibit the growth, they are classified. So first class is antibacterial antibiotics, means that antibiotic or that chemotherapeutic agents which kill or inhibit the growth of bacteria. Depending upon the two properties, further antibacterial antibiotics are subclassified as bactericidal antibiotics and bacteriostatic antibiotics. As the word, it is the antibiotics which are bactericidal antibiotics, means these antibiotics kill the harmful microorganisms. Examples are penicillin, cephalosporin, aminoglycosides, etc. Whereas bacteriostatic means that antibiotics which inhibit or just prevent the growth of bacterial species. Example is tetracycline, chloramphenicol and clindamycin. Next antibiotic example or classification is antifungal antibiotics. Examples are griseofulvin, amphotericin B, nystatin. Next class is anthelmintic antibiotics or anthelmintic agents such as ivermectin. And last class is that anti-cancer antibiotics. Example is bleomycin, dactinomycin and doxorubicin. The next class, depending upon the bacterial spectrum of activity, depending upon the spectra against which the antibiotics show act on the different species of microorganisms, they are classified as broad spectrum antibiotic and narrow spectrum antibiotic. Broad spectrum antibiotics are that these antibiotics inhibit the growth of wide range, that is more than one species of microorganism. The example of these antibiotics such as cephalosporin, chloramphenicol, tetracyclines, etc. Some antibiotics can inhibit both, that is gram positive also and gram negative bacteria also. Whereas narrow spectrum antibiotics effective mainly against a single species of microorganisms, either may be gram positive or gram negative bacteria. Example is bacitracin, nystatin and penicillin. The next class, which is depends upon the mechanism of action. The first one is cell wall synthesis inhibitors. Examples are penicillin, cephalosporin, carbapenems and azithromycin. Here, the antibiotics act on the cell wall synthesis procedure and the precursors which will leads to the cell wall synthesis, and the cell wall synthesis is going to be inhibited and it will shows the killing of or destroying of the microorganisms. Next is microbial cell membrane inhibitors. The cell membrane which helps in the cellular processes or which will protect the cellular processes that are going to be inhibited. So example is polymyxin B and E, colistin, choline and imidazole. Next class is protein synthesis inhibitors. That proteins which are involved into the cellular processes are going to be inhibited, and that's why the microorganisms growth are going to be inhibited, or that microorganisms are directly going to be killed. Examples of antibiotics such as aminoglycosides, macrolides and tetracyclines, etc. The next class, which is depends on the chemical structure of antibiotics. This is the most important classification of according to the medicinal chemistry, where we consider the classification depending upon the basic moiety or the basic structure present into the antibiotics. So depending upon that, the first class is beta-lactam antibiotics. Here the examples are penicillin, cephalosporins, carbapenems and monobactams. Here the main moiety is the beta-lactam ring. The next is aminoglycoside antibiotics. That antibiotics which contain glycosidic groups with amino group. Example is streptomycin, neomycin, kanamycin, gentamicin, tobramycin and amikacin. Next class is tetracyclines, as word indicate, that contain the 4 cyclic rings in a fusion. That is, the examples are tetracycline, chlorotetracycline, oxytetracycline, doxycycline, minocycline, metacycline and meclocycline. The next classification is macrolide antibiotics. That means that antibiotic moiety or the main basic structure is a large structure. So example is erythromycin, clarithromycin, azithromycin and roxithromycin. The next class is polypeptide antibiotics, means that antibiotic which contain number of or more than 2 or 3 polypeptide linkages. Example is actinomycin, bacitracin, colistin, polymyxin B and tyrosine. The next class is polyins, means that antibiotic which contain more than 10 double bonded structure or double bonds into the structure. Example is amphotericin B and nystatin. Next class is lincomycin, example is clindamycin, and the last one is miscellaneous antibiotics. Examples are chloramphenicol, novobiocin, mupirocin, fusidic acid, etc. So these are the different classifications, and most important classification is based on the chemical structure of all antibiotics. So we stop here. Thank you for watching the video. Stay at home, stay safe and study hard. Thank you very much.